Okay, let's have a class.
I guess.
So today we will do pole plating. Before that, let's look at your results. I did the 4 labs results right here. You guys should have your own plates, which is on your tray. And you can take a look at what you have. So what we did. So I will first say review and count CFUs for spread plating. So remember how we did it. It's right there. I did not remove it because we can use it. Is that right? That's exactly what you did. What do you have? You have 3 plates right here. And I said very clearly, your label has to be very good, and we're going to label the dilution factor, which is the final dilution factor of the plates on the agar. And remember, at the back of the agar. So what you have, this is 10 to the power of 3, 10 to the power of 5, and 10 to the power of 7. You look at you, 10 to the power of 3 plates, what it looks like. Does it look like this? A big mess. Is that right? You cannot even see a single colony there. It's kind of like a messy. Or a big chunk of cluster. Now can we count? We cannot count. So what we say? Recording TNTC. This equals 2 numerable to count. Sometimes we also say TMTC, too much to count, too numerable to count. Okay, look at you 10 to the power of 5. Is that looks like this? You started to see some of the single colony, but you still cannot count. It's still uncountable. Is that correct? Look at your own plates. So that recording still TNTC. You can take a picture, stick on your notebook. Then look at the 10 power of 7. Did you do like what I did? Spread very well. And E. colony should be very huge. And remember, E. coli is mesophile. At 22 or 25 degrees Celsius, 5 days can grow very good. Does your place look like that? That's another plate. That's another lab, what I did. And this is the last lab, what I did. Did you guys do a good job? What means a good job? Which means 10 to the power of 7, you very clear have colony there. What we say? Colony forming unit, CFU, represents colony forming unit. Okay, what you're going to do then? The next step is counting. How did you do the counting? Let's have a Sharpie marker. Who had a Sharpie marker? I'll bring it. Yeah, you have this Sharpie marker as an example. So how you counted it? Well, it seems like easy. You learned this in your kindergarten, how to count apples or oranges. So how you counted it. Some people count like this. Or some people just like this. Oh, 1, 2, 3, 4, 5, 6, then maybe forgot what we should do. This is the reason why the back is important. When you see one colony, use Sharpie marker. When you count, mark it. The second count, mark it. Then you mark it. Every time you count it, you mark it. Okay? So what you do is you're gonna count it. Okay, whenever you count, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So like that. And I believe most of you, the 10 to the power of 7 plates is fit the countable zone, which is 30 to 300. Now, how we do the calculation? Let's say your final number is 30. This is CFU on agarose. And what is the dilution factor? 10 to the power of 7. This is dilution factor. And remember, we talked about that. This is the final dilution factor of the plates. And it is going to be 10 times more than the dilution factor of the tube, because we add 0.1 milliliter there. So how many we get? We get 3 multiplied by 10 to the power 8 CFU. And you have to remember, this is a unit per ml. So I have to be cautious to tell you what we did here is the concentration. It is not all the bacteria in this tube. If we want to see the whole bacteria in the tube, you need to multiply by 10 ml. What we did in spread plating, we always do is the concentration. Okay, and the concentration is 3 multiplied by 10 to the 8 CFU per ml. Now, in the lecture, we mentioned about that. Because this is a huge number. And lots of the times, we don't compare these huge numbers because they will have an error. The error is too huge for the statistics analysis. So what we need to do, we need to transfer this to the log. Is that right? Log 10. So, 3 multiplied by 10 to the 8 CFU is how many log? This, if you transfer to log, equals 8, is that right? Now you have a 3. How many log? You can do the calculator, about like 0.2. So that will be roughly 8.2. Okay, so we do other ways. We do log 10 to the 8. We get 8.2 log 10 CFU per ml. Which means this number, you need to transfer them to the log 10. That means 10 to the power of 8.2 approximately of 8. Remember? Is that good? Math. You can use calculator. You don't have to do it. I just know it. Approximately, not exactly. Does anybody, everybody's clear? We got this number. It's too large. Bacteria is huge. This is 300 million. Like in the lecture we mentioned about, if we have so many data like this, we cannot do the comparison because the error is too huge. So we transfer this to the log units. We become a log. So we do log 10, bracket 3 multiplied by 10 to the power of 8. Then you do the calculator, you get 8.2. That is something like 10 to the power of 8.2 equals approximately 3 multiplied by 10 to the power of 8. So, what that means? Which means this tube, the concentration is 8.2 log CFU per ml. Okay, you can do based on your own results. use the way what I teach you, and then you calculate and put on your notebook. That is a spread plating. Now, regarding the dilution factors, on Thursday, we will do is a review for the midterm exam. Midterm exam is next week, Tuesday. And we also going to talk about the dilution quiz, because that's you take home and you and your bench partner work together to do the dilution quiz, okay? So we will be doing that. We'll be introduce that on Thursday, and also we're going to do the review. Okay, what we're going to do today? We will do pore plating. So why I did not remove majority of these here? Because they are the same. Now pore plating will be chapter 11. So first of all, let's just review what we did in the last time. We have two different types of the dilution, which is 10-fold and 100-fold. Now what's the difference between that? 10-fold we're using 9.0 ml dilution tubes. And we add 1 ml each time. A 9.9 ml is used for 100 dilution factor, dilution schedule. And instead of we add 1 ml, we add 0.1 ml. And always remember, this is tube dilution. The final you added will be the final dilution factor. They're 10 times more than the tube dilution. So what we're going to do today, it's exactly the same, except we don't do spread plating, we do pore plating. Now what's the difference? Spread plating, we're using the agar. It's already being measured. And pore plating is we are going to add the liquid onto the empty petri dish. And then we add the melted agar. The melted agar is at the water bath. And I took 2 bottles out because it's too hot there. It's inside about 60 degrees Celsius. Remember, you had a practice question in the lecture. Pore plating, usually we get a lower number because of the mild heat gonna killing bacteria. Remember that question in your review questions? So that's why I took 2 bottles out, let them cool down a little bit. Okay, so how are we going to do? The same thing, you get your tube, okay, that's a bacteria there. And then you get your 3 empty tube, 3 dilution tube, which is 9.9 ml, 0.1% nutrient broth. You add 0.1 ml from the original tube, go to the first one. Shake very well, go to the second one, and go to the third one. That is called serial dilution. It's exactly the same as you did for spread plating. Now the difference is here. We got to do the same labor. Okay? And we also going to use only 1 tip to add it there. The difference is we are adding 1 big droplet, microliter. Here, microliter. Here. Then we will go every bench to pour for you. Okay, the melted agar will pour for you. How many we pour? Approximately 20 to 25 ml. It's a melted agar. So basically, is this agar plates, you add your solution there. That is 100 microliter diluted bacteria solution. And we're gonna pour melted agar, approximately 20 to 25 ml. And then you do a very gentle stir. A very gentle stir to mix it. At room temperature, the agar will be solidified relatively quick, about like 3 minutes, it will be solidified. And again, you put on your tray. And we'll wait for 2 days, we'll see how it looks like. That's the way we gonna do. Okay, 2 days, 48 hours, we should be good to see some colony. Look at the 5 days. That's very huge. The colony looks huge. So 2 days we should be good, 48 hours. Again, only one tips for the dilution, but we don't use spreader. So we add it into the empty petri dish and then we do. So, what are you guys gonna do? This will be for 2 days. Everybody go ahead to get 3 empty petri dish, 3 dilution cubes, and get the bacterial solution. Go ahead to do the cereal dilution until you add here. You have your hands on and we go to help you pour the melted alcohol. Okay? That's what we're going to do. But basically all the same.